super seminar series. Now, the land acknowledgement. We acknowledge the land the OMNI-RÉUNIS Network's head office is located on York University. Recognizes that many indigenous nations have long-standing relationships with the territories upon which York University campuses are located that precede the establishment of York University. York University acknowledges its presence on the traditional territory of many indigenous nations. The area known as Tikaronto has been caretaken by the Anishinaabe Nation, the Haudenosaunee Confederacy, and the Huron-Wendat. It is now home to many First Nation, Inuit, and Métis communities. We acknowledge the current treaty holders, the Mississaugas of the Credit First Nation. This territory is subject to the Dish with One Spoon, One Poon Belt Covenant, an agreement to peaceably share and care for the Great Lakes region. Now, a little bit about the Super Spreader Seminar Series. Now, this seminar series encourages highly qualified personnel. And so we're talking about postdocs and graduate students to present their research, then also to discuss the ideas and share research experiences and establish connections among various branches of the OMNI network. It is also a platform to spread scientific knowledge about One Health research framework and showcase research progress of the 30+ OMNI HQPs within the network scientific research community. And so this seminar is supposed to be beneficial for knowledge transfer among OMNI and the emerging infectious disease modeling networks and collaborating scientific communities. Now we have for the speaker for today, Dr. Shivdeep Hayer. And Dr. Shivdeep Hayer is a veterinarian researcher with an interest in infectious disease research. He had his PhD dissertation research at the University of Minnesota, focused on utilizing diagnostic laboratory data to evaluate antimicrobial resistance in pathogens collected from pigs. Now he's continuing his research on antimicrobial resistance at the University of Guelph and is currently analyzing aspects of antimicrobial resistance surveillance data generated by the Public Health Agency of Canada. And so at this point, I would welcome Dr. Shivdeep Hayer to continue with his talk for the day. Thank you.
Okay. Share my screen. Okay. Can everyone see the screen? Yes. Okay. Good morning, everyone, and thank you for giving me this opportunity to deliver this seminar today. Today, I'll be talking about changes in antimicrobial resistance in North American pigs. This is a work that I did at University of Minnesota for my PhD dissertation, and I'm currently working at University of Guelph, working with the Canadian data. And the stories do overlap a little bit, so I decided to merge them and tell you something about the lessons that I learned along the way. So a brief introduction to antimicrobial resistance. As defined by the CDC, antimicrobial resistance is the ability of any microorganism to stop an antimicrobial from working against it. Now that microorganism could be a virus, could be a bacteria, could be a fungi. However, today I'll be talking about talking in the context of bacteria only. And antimicrobial resistance is an emerging public health issue, especially over the last 2 decades. And some experts have suggested that antimicrobial resistance is expected to cause nearly 10 million human deaths per year by 2050. However, bacterial pathogens, animal populations, and animal production systems, as well as policies regarding antimicrobial usage, they constantly evolve. Hence, this threat of antimicrobial resistance is a constantly evolving problem. A brief history of antimicrobial discovery and resistance. Here I have provided a graph which shows when a particular class of antibiotic was discovered and how quickly did the reports of emergence against this particular antibiotic class showed up. So as you can see in the large blue on the right hand side is penicillin. So penicillin was discovered in 1928 and the reports of emergence started coming out in 1940. In fact, Alexander Fleming, during his acceptance speech, has mentioned that this emergence of penicillin resistance will be a big issue in the coming years. In 1948, tetracyclines were discovered, and emergence of tetracycline resistance was discovered in 1953. As soon as a new antibiotic is discovered, the resistance against this particular antibiotic class emerges quickly too. And also a point to note here is that it has been 30 years since a new class of antibiotics was last introduced. So we are running out of options when it comes to antibiotics. And hence, the aim of surveillance is to basically conserve the antibiotics that are currently effective. Hence, the World Health Organization have classified antimicrobials on the basis of importance, and they have classified certain antimicrobials to be critically important or highest priority. And among those are cephalosporins and quinolones that we will be talking about a little bit more. Tetracyclines and sulfonamides are the older classes, and they are no longer the high priority antimicrobials. So that's something to keep in mind. Epidemiology of antimicrobial resistance is extremely complicated. It's not linear. So there are several ways, several pathways through which antimicrobial resistant bacteria can transfer from food animals to humans or from environment to humans. So it's definitely not a linear path at all, which complicates any modeling efforts any modeling or surveillance efforts that we put in practice. When it comes to animal production, this statement was given in 2001, and it actually created a lot of ruckus back in the days. It's that it was estimated that 70% of antibiotics produced are used in food animal industry. However, the authors did not mention the particular antimicrobial classes and how these antimicrobials are used. So antibiotic use practices vary across different animal production types. So the antibiotics administered to poultry might not be the same as being administered to swine, for example, or your pet animals, dogs and cats. Also, antibiotics are used for several purposes, from growth promotion to treatment. And it's usually at the growth promotion level where the policymakers are becoming a lot more stricter. And antibiotics can still be used for treatment, although the regulations are becoming stringent for the usage, even for treatment purposes. So the current antimicrobial resistance surveillance in USA and Canada is based around two programs. One is NAMS, the National Antimicrobial Resistance Monitoring Surveillance, and that's in USA. And CPARS, that's a Canadian Integrated Program for Antimicrobial Resistance Surveillance, that's in Canada. And both of these have an active surveillance component, and they do, at least when it comes to pigs, they survey meat samples, that is the pork samples, market pigs, and CPARS and the Canadian AMR surveillance system also samples the abattoirs and farms. NAMS also has a VET LIRN program, which is a passive surveillance program, and their focus is more on bacterial pathogens from animals or from diseased animals, but it's still in its infancy, and it's not really grown that much. It only started in 2019. And the focus of all these antimicrobial resistance surveillance programs obviously is public health. Another source of data that we have and which can be utilized and which I did utilize for my research is passive surveillance using the veterinary diagnostic data. So a lot of veterinary diagnostic labs, such as University of Minnesota, University of Guelph, University of Montreal, they do have their veterinary diagnostic labs, and they do passively receive a lot of submissions from swine farmers or swine veterinarians because they want to get a diagnosis. So they end up collecting a large number of data, particularly on bacterial pathogens from swine or from poultry. And at least at University of Minnesota, we received samples from all major swine producing states, because University of Minnesota and Iowa State University are two of the biggest swine programs in USA. So we did receive samples from all the major swine producing states. However, the issue with these diagnostic samples are that these diagnostic samples and the bacteria isolated from these samples might have been directly under selective pressure because of previous antimicrobial use. So what really happens is that veterinarians primarily try to treat the animals at their level, so they do use antibiotics. However, if the problem persists, it's then that they send in samples. So there's always this issue of them being exposed to some antibiotics. So this was the flowchart for my PhD dissertation. I conducted a systematic review of global phenotypic and genotypic prevalence in pigs. I studied phenotypic AMR or antimicrobial resistance in USA using diagnostic data. And then I sub-selected a few isolates of interest and did the genetic characterization of them. So I'll be using terms like phenotypic and genotypic a lot. So phenotypic is something that you would see in your petri dish. And here you can see that certain antibiotic discs. So these white dots are your discs, and we have created a plate of bacterial growth in the background. And here you can see that the big black halo or big black halos are basically indicate that that these antibiotics were effective against the growth of the bacteria in the background. So this is something that you can see. It's called phenotypic. Whereas the genetic characterization is more at a bioinformatic level. We don't really see the bacteria, but all we get is a code of 80 GC nucleotides. So topic 1 was antimicrobial resistance changes in clinical E. coli isolated from diseased pigs between 2006 and 16 in the US. So time of analysis was 2006-2016, and we had nearly 10,000 E. coli isolates. We had a standardized method, and which was equivalent to the method used by CLSI, which is also the method that's used by NARMS and CPARS. And the level of analysis was unique isolates at a farm month level. So here you can see it's a pretty busy graph, but I want you to just focus on this violet dashed line at the bottom. So this is the line for fluoroquinolone resistance or enrofloxacin resistance. And here you can see that the enrofloxacin resistance is nearly zero in 2006. However, it increased gradually and drastically. And by the time it was 2016, the resistance percentage was up to 20%. So what happened was that in 2009, enrofloxacin was introduced in swine to treat respiratory pathogens. And 2012, it was also allowed for enteric pathogens. Hence, enrofloxacin was used a lot more, and we were able to see this trend quite clearly. I would also like to point out this yellow dashed line, that's for extended spectrum cephalosporin resistance. And as I have mentioned previously, it's a highly critical antimicrobial class. But at a phenotypic level, the percentage of resistance persisted between 30 to 40%. There was not a clear trend at all. And lastly, the big red line at the topmost, it remained consistent, but nearly 95% of the isolates were resistant to tetracycline. That's this solid red line at the top. So these were the few big changes, and I would talk about them at the genotypic level also. So the interesting thing about this enrofloxacin resistance that we can clearly see here is that till 2014, 2015, we were not able to see this increase in enrofloxacin resistance in the data collected by the NAMs. So the NAMs data is collected from food animals or pigs who are ready for market or ready for slaughter, or from pork or meat sample. However, after 2016, if you look at the current reports generated by NAMs, this trend of increased fluoroquinolone resistance or increased enrofloxacin resistance has started to show up in the NAMs data also. So I guess the point that I'm trying to make here is that the clinical data, although it's actively collected, it has its own biases, can actually be a very early stage indicator of what's going to happen later on in the food samples that we are ultimately going to consume as humans. Let's just repeat of what I have mentioned. I also created antimicrobial resistance networks, and here you can see in 2007 the network was a lot more sparser, and in 2016 the networks of different antimicrobial resistances became a lot more denser. So this can be an indicator of either the isolates becoming more resistant or resistant to a large number of antimicrobial classes, or they becoming more pan susceptible. However, if we collaborate this data with the previous data we have shown, this is more likely an indicator that the multi-drug resistance was increasing over time, which is again a cause of concern, because from a clinical perspective, what this means is that that bacteria would become resistant to a large number of antimicrobial classes, hence the likelihood of antimicrobial being effective will be less. So I subselected isolates belonging to extended spectrum cephalosporin, and I wanted to see what was happening at a genetic level. And as I have mentioned in the previous slides, that the level of antimicrobial resistance to cephalosporins actually remained pretty much constant. However, at a more genetic level, we were able to see some differences or difference in emergence of genetic mechanisms. So just a brief background of what are the occurrences of antimicrobial resistance at a bacterial level. So resistance can emerge in three different ways, or two different ways. One is there might be mutations at a chromosomal level, which can increase or decrease the efflux pump activities, or create mutations in the proteins, or create alterations in the proteins. Or there could be plasmids. Plasmids are extra chromosomal, so they are not part of the bacterial nucleus, but they can be exchanged between bacteria, and they can carry antimicrobial resistance genes, which can spread from one bacterial family to another bacterial family, or from one bacterial pathogen to a commensal bacteria. So the research questions I had here were, what genes are responsible for these resistance? On what kind of plasmids are these present? Are E. coli isolates resistant to cephalosporins more closely related to each other? That is, was there a clonal spread, or were they vastly different? That is a horizontal spread. And how similar are these E. coli in diseased pigs to those isolated from other animal species, including humans? So I had selected 212 E. coli isolates between 2014 and 15. I did sequencing on these isolates. And here you can see the prevalence and geographical spread of cephtifer or cephalosporin resistance genes. So the BLA-CMY2 was the most dominant genetic mechanism for cephalosporin resistance, and nearly 84% of the isolates carried this genetic mechanism. However, what's interesting is that if you compare these isolates collected in between 2014 and 15 to the isolates collected between 2008 and 9, in 2008 and 9 in the US, all of the cephalosporin resistance was due to BLA-CMY2. However, in 2014-15, we have also seen the emergence of other mechanisms, particularly the BLA-CTXM types over there. And this is also a pattern that we have seen in the Canadian context also. So although at a phenotypic level, the prevalence of cephalosporin resistance remained consistent, at a more finer mechanistic level, we were able to see changes happening. I created phylogenetic trees to basically see whether there was an association between particular E. coli type and particular genetic mechanisms. And here I have classified as 4 different clades. I'm not going to go deeper into it, but the bottom line here is that there were certain E. coli subtypes that were associated with certain genetic mechanisms. And here I'm showing that even within a particular E. coli, so we have E. coli, and E. coli can be differentiated into several different subtypes, or we call them ST. And even within these subspecies or subtypes, there can be further differentiation. So here I have, from the previous graph here, I have extracted or have zoomed into ST744. This is an E. coli type. And even within ST744, we can subclassify them into 3 different clades, which are labeled in yellow, purple, and green here. And here I've also mentioned that, so this was a comparison between the isolates in our study to the animal isolates and human isolates globally. And here we can see, if we just focus on the yellow one, we had a couple of isolates that were present in our database, which were also present in humans in the US, as well as humans in Netherlands and Philippines. Similarly, if we look at the violet labeled or violet coded or purple coded, I'm not good with colors, or the purple coded clade, we have isolates from diseased pigs in US, but they're also very similar to isolates collected from healthy pigs in the US, as well as healthy pigs from cows. So I guess the bottom line that I'm trying to present here is that we have this E. coli subtype carrying particular genetic mechanisms to resistance, and they are spread across different animal species, regardless of the disease status, as well as there are some public health implications because they are very similar to isolates that have been found in humans globally, which was, I thought, was a pretty interesting catch. Then I further zoomed into those plasmids that I talked about. And here's a plasmid that I'm focusing on. And so there are 2 interesting things happening here. One is that this particular plasmid is carrying a very large number of genetic mechanisms and which can cause resistance to several antimicrobial classes. So the BLS-SHV2 can confer resistance to cephalosporins. SUL, SUL2 or SUL1 can confer resistance to sulfonamides and so on. Another interesting thing is that this plasmid that we found in diseased pigs in US was actually very similar to isolates present in Taiwan and present in humans in USA as well as humans in Denmark. So that was my conclusions here, that we actually identified some novel plasmids. Novel, they were not necessarily novel because they were very similar to existing plasmids. We also reported some new genetic mechanisms, and these isolates both at an E. coli level, at a chromosomal level, as well as at a plasmid level, were similar to what has been found globally. And this is the reason why it's called the antimicrobial resistance phenomena, or the problem is considered to be a silent pandemic. And this is an example of that. You are seeing plasmids that are present globally. However, we do not exactly know how these plasmids have been able to jump from one bacterial species to another bacterial species, or from humans to animals and vice versa. So coming to the Canadian aspect of the talk. So what I'm doing right now at University of Guelph and with the OMNI network is we are focused on the emergence theme. And what I'm trying to study is, are the antimicrobial resistance genes and bacteria similar across different food productions systems, swine, poultry, and dairy in Canada? And this is very similar to what I have already shown in the previous slides. But I just want to replicate the same study that I've conducted in the US in a more fine-tuned way in Canada. And do these genes and bacteria vary across different geographical locations within the same animal species? And on top of that, are there any environmental factors that are at play? So as I mentioned, there are linear models of AMR risk assessment. We have a pig which is ready to be slaughtered. It goes to the slaughterhouse and ends up on our plate. However, what we are trying to establish is we are trying to see what are the how these antimicrobial resistant bacteria transfer between different food production types. So what's the relationship between antimicrobial resistant bacteria isolated from poultry with swine? Also, we are trying to see if the antimicrobial resistance that bacteria that we see in Ontario, is it similar to what we see in Quebec or in the prairies? So we are leveraging the data that has already been collected by the CPARs and the FoodNet. And we are also leveraging the expertise of GRDI, which is a Genomics Research and Development Initiative. These all different programs fall under Public Health Agency of Canada. So I started with the phenotypic E. coli data that has been generated by CPARs and FoodNet. So we have nearly 14,072 E. coli that have been isolated between 2008 and 2021, collected from 190 farms across Ontario, Quebec, Alberta, Saskatchewan, and Manitoba. And between 2008 and 14, CPARs used to collect 3 isolates per sample, and after 2014, CPARs only collected 1 E. coli isolate per sample. So that's another issue that we are currently facing. So at a broader phenotypic level, these are the changes in E. coli resistance that we have observed in pigs over the time. And we have some good news in some way. At the topmost, similar to what we have seen in the American data, the most tetracycline is the most prevalent resistance out there, or resistance to tetracycline is highly prevalent in E. coli. However, over the years, the resistance to tetracycline has actually, so this is this topmost line in pink, has decreased from 80% to nearly 65%. Similarly, sulfonamide resistance has also decreased from, and that's this in light purple, has decreased from 42% to about 37%. So those are the two big changes that we are seeing, both trending towards a decrease. We have also observed differences in E. coli isolates collected from different regions. So here I'm focusing on tetracycline resistance, but stratifying it across different provinces. So the red line here is the national level data, so everything combined. The trend of E. coli resistance on a decline is what we have seen across all different provinces. However, the prevalence of resistance has always been lower, significantly lower in the isolates collected from the prairies. That's this light blue line at the bottommost. Whereas the isolates collected from Ontario and Quebec have always been more resistant to tetracycline. So there's also a difference in resistance patterns across space or across geography. So another thing that I was focusing on was whether the isolates are becoming more resistant or less resistant to a number of classes. So whether they're becoming multi-drug resistant. So the pattern that we are seeing here is that if you look at the line in red, that's your pan-susceptible isolates or isolates resistant to 0 antimicrobials. And the trend is that these pan-susceptible isolates are actually becoming more highly prevalent, which is again a good news. However, conversely, we are also seeing that the bottommost line, that is the E. coli isolates that are resistant to 5 or more antimicrobial classes, their prevalence is also on an increase. So what we are seeing here is kind of a forking pattern where either the isolates are becoming more pan-susceptible or they are becoming highly resistant. And this is something that I would like to focus more at a genotypic level, because it would be interesting to see whether there are multiple mechanisms of resistance being present on the same plasmid or not. So this basically serves as a preliminary data for that. I also used clustering methods, differentiated them into clusters, and again got the same result. The topmost one is the E. coli isolates that are either resistant to 1 or resistant to 0 antimicrobial classes, and their prevalence is on an increase. And the D, the bottom here, is highly resistant antimicrobial clusters, and their prevalence is on an increase. The prevalence of this topmost one is also on an increase. That's the prevalence of pan-susceptible isolates is on an increase. So getting the same results as we saw in the previous slide. On a network approach, we are also seeing that if we compare the networks created using antimicrobial resistances, these networks are becoming denser over time. So if you see in 2018 to 2021, that's on your right-hand side, both in terms of degree, in terms of connectivity, different other social network metrics, the networks are becoming more denser, which could mean either they are becoming more pan-susceptible or they're becoming more resistant. And we would like to fine-tune this. I would like to see what's happening at a genotypic level. So we are in talks with the CPARs to have access to the genotypic level, to the genomic data also. So Salmonella data in brief, I'm not going to go too much into it, but the only worrying trend that I saw here was an increase in gentamicin resistance and an increase in cephalosporin resistance over time. However, the prevalences are still extremely low for these antimicrobials. These would be all green lines at the bottom, essentially. We saw pretty much a similar story as we saw in E. coli. The pan-susceptible isolates were on an increase overall. So was the isolates that were resistant to a higher number of antimicrobial classes. So overall, I would like to conclude that there are statistically significant changes in AMR in pigs over time in a Canadian context. And there are changes in multi-drug resistances over time, and we confirmed that using different statistical approaches. We would like to further refine and get to the root cause of these changes that we are seeing in antimicrobial resistance by incorporating metadata such as serovar, antimicrobial usage, farm-related factors such as biosecurity being used. And if we incorporate all these different data with the AMR prevalence data, we would actually be able to see what led to a decrease in tetracycline resistance over time, and as well as why is there more tetracycline resistance in Ontario and Quebec as compared to the prairies. So future directions, analysis of CPAR's data from other sources. So I would also, going back to the topic of integrating AMR from different sources, I would also take a look at the poultry and beef data, and I've already mentioned the genomic analysis. So with that, I would like to acknowledge my team in USA, the CVM VMAT program at University of Minnesota, and my collaborators in Canada, and the OMNI network and the Public Health Agency of Canada. So with that, I would like to take questions. Thank you.
Okay, thank you, Shiv, for the talk. Yeah, that was a great talk. Yeah, so I don't see any questions in the chat yet. So anyone who wants to ask a question or pass a comment can do so right away. You could raise your hands and I'll call you, or you can unmute yourself and ask your question. Okay, I guess there are no questions. Well, we'll still give people like 1 minute. If someone has a question to ask, yeah, you can go ahead and unmute yourself. Okay, I still don't see any raised hands and there's no question in the chat. Yeah, so I guess, and it's almost time, so I guess we will end the meeting for today. And thank you so much for attending, and thank you, Shiv, for the talk. Yeah, that was great and interesting. Okay, thank you.